Sarah. I'm a PhD candidate in the Morris Laboratory here in the biology department at UAB. Today I'm going to be walking you through how I make Agar art. I'm also an artist at a gallery downtown called Ground Floor Contemporary, so this is a lot of what I do to inform the public about the work that I do here in the lab. But we also use it as a really cool educational tool, not just here at UAB, but across the country. I work really closely with a professor, Dr. Erin Arnold at Jeff State Community College, and her students go through a similar curriculum as our students do, where they use Agar art as an investigatory tool to learn more about bacteria that they've been working with throughout this semester. So I'll explain more about why bacteria completely fascinate me in a second, but first I want to show you how we actually do Agar art. So first, like a good scientist, I'm going to clean off my bench with alcohol to disinfect anything that might be crawling on this bench. And then wait for it to dry so that I do not light the lab on fire. That would be bad. Alright, and then I'm going to turn on this Bunsen burner. First time, I've never done that before. Okay, just kidding, I have. So what this is the flame acts like a sterile orb to decontaminate anything that could get on the plates that I'm trying to work on. So that's why it's really important that when we're working with bacterial cultures or anything else, that we stay around the flame, because we don't want our plates contaminated, we don't want ourselves contaminated, we just need to be hyper aware of safety. You could also work in a hood, but for this demonstration I'll just be working around a flame. So here I have a petri dish. So this is a piece of plastic that has a lid in a circular shape. What it's filled with is nutrients that bacteria need to grow, and these are very similar nutrients to what we need to grow. There's carbon, phosphorus, nitrogen, oxygen, the same things they need to grow, same things we need to grow, except it doesn't look quite like a hamburger. It's also full of a gelatinized substance that's a lot like hardened jello. This will allow us to work with the bacteria in the ways that we need. Something really cool about that gel is that it comes from a different microbe, but that's a lesson for another day. So I have a blank petri dish, and I have 3 different cultures. Here I have Pseudomonas aeruginosa from my colleague Melissa Walker. Don't tell her I'm stealing this. Melissa, if you're watching, I'm sorry. I have another culture that's growing up, Serratia, and this is, I'm pretty sure it's a pathogen. It was collected by one of the students in our classes, and they're still working to figure out what it is. So we will stay tuned on that, and I will tell you how they did that, how they got it in a minute. So I'm going to take out a wooden, I got an extra stick. These are sterile wooden sticks. You could also use, if you wanted, an inoculating loop, but because they're a little bit more bendy, I prefer to use the sticks. And so I'll use them once, and then I will put them to be autoclaved, and I keep on reusing them after they have been sterilized again. So here I have a drawing. UAB, go Blazers. I was going to do Roll Tide, but then I thought better. So if you can see closely on this plate, there's a bunch of lines and a bunch of dots. What this is a diluted culture over here. So when you're preparing these from a stock culture, you spread a lot of bacteria on here, and then you take a new stick or a new loop and do it again. And then you do it again, and then you do it again. And then when it grows up, you have these dots, which we're assuming are, like the entire colonies are free from contamination, and we can go ahead and just pluck some bacteria from one of those colonies. So it's a way to get a lot of dense bacteria isolated for our use. So we're going to take a colony of bacteria. Okay, I already got one, but let me get a little bit more. I'm going to go over here to my plate, being very careful, and I'm going to trace my image onto the agar. And then I will use a new end of the stick. Alright, so then I made the outline of UAB. But what's really cool about what we do in our classes and with other students is we have them use multiple bacteria. That way when their artwork grows up, you can see very unique interactions between the bacteria that you wouldn't just see if you were doing normal biochemical tests. So I'm going to take a new sterile stick. Give me some of this gross dude. And then fill in my UAB. I think it was his name, the student who prepared this, Brandon Harris, who took microbiology last semester. Their group named this lava lamp because when it was growing up in liquid culture, it reminded them of a lava lamp. Actually, I think I have it right here. It's pretty spooky. What? Oh. Okay, so here we are. Filling our UAB with red, just to appease my dad because he likes Alabama.
Question: why did you trace the UAB?
Why did I trace the UAB? So now you can see on the dish, you see the drawing, right? Kind of. So when it grows up, these take a couple days to grow. You will see the entire image. Does that answer the question? And I traced UAB because I like UAB. So you can't see it yet because the bacteria haven't replicated. So in each of these colonies that I picked from, there's over 1 million bacteria, and then I took them out of that culture and I put them onto this plate. But what they still need to do is divide and grow, so that when we actually see the image, there's going to be millions more bacteria than when I started. They reproduce by binary fission, which is sort of clonal, unlike how we reproduce sexually. So we expect that when this grows up, we will see the same types of colonies as the colonies that we laid down, or the same types of colors. Okay, so how did we get this bacteria? This is probably the coolest thing in the entire world. So, this is a bag of soil. Actually, you can get bacteria from almost anywhere, but in our class, we have students work from soil. Okay, bacteria are everywhere. How many bacteria do you think are in a teaspoon of this soil? Hundreds. Hundreds? That's a good guess, Kim. Maybe think a little bigger. So trillions. Up to 1 trillion bacteria are in 1 teaspoon of soil. This is like blowing my mind. So that's the same as if you put more than the US population on UAB's campus. Like imagine how crowded and packed that would be. Like think on that scale. That's how many little bacterial creatures are in a teaspoon of this soil. So what we can do is take a little bit of the dirt out of a bag and put it in some water or saline and shake it up real good, and then we can take a portion of this out of the dirt water basically and plate it on a new plate. So it'd be like if I just took a small amount out of here and put it on here and spread it around real good and then let it sit. When I come back a few days later, I'm going to see all sorts of different cultures that are different colors representing different species of bacteria. So then what the students do is they look at their plates and they're like, oh, I like the prettiest one. In this case, they liked the one that looked like this. And then you can take it and isolate it and make a streak plate just like what they've done here. And they'll use these bacteria throughout the entire semester, and they will identify them using lots of biochemical tests, but also they'll look at their genes, their DNA, through sequencing technologies. Unfortunately, they haven't quite figured out what this one is. It's pretty elusive, pretty tricky. So we can do that. We can culture a lot of these bacteria, but something that's also important to realize is that there's a lot of bacteria that are in this dirt that we can't culture with all of these petri dishes and with these methods. That's why researchers use other types of analyses to figure out all of the species that are in this dirt. So through next generation sequencing, they can actually use those technologies to map out all of the DNA that's in this community. And what that allows them to do is see the hundreds of species that we wouldn't normally be able to see using the petri dish methods, which is really cool. It's also the same technique that medical researchers use when they're analyzing the human microbiome and all the bacteria that live inside of us. Something that amazes a lot of people who don't know this already is that those researchers, in the same way that we look at soil, actually look at human and mouse poop. So They collect the feces of mice and humans and compare them, and they can look at all of the microbial communities that are in those poop samples as a way to figure out all the bacteria that are living in our gut. So we're using the same technologies here in our lab, except we're looking at soil bacteria. I'm really interested in how antibiotic resistance spreads in soil communities. Something that's really cool about this type of artwork is that it will show us interactions between bacteria, like antibiotics. So antibiotics are chemicals that bacteria produce to fight off other chemicals. Actually, that's contested. Some scientists think that they're just signaling molecules, but that's a whole can of worms. Again, not going to get into that. But they do function as a way to fight off other bacteria. And what Alexander Fleming did in the 40s, and some of his contemporaries, is they figured out how to harness that chemical so that we could use it in hospital and clinical settings. But all of the while, under our feet, bacteria are still fighting with these chemicals, whether or not we're using them in the hospitals. And in fact, a lot of our overuse has selected for more antibiotic resistance in bacterial communities. Because we're pumping all these chemicals at a sort of unprecedented rate, bacteria are evolving and responding in ways that can be really important for us down the line, and are even continuing to be important for us. Antibiotic resistance bad. Yes. Bacteria are really cool. They live in your nose, they live in vents, they live all over the world. And I think it's really cool that we get the opportunity to study them and to actually look at things that we can't normally look at. Yeah. Are there any other drawing ideas that I can do aside from good old UAB? Yeah, a question?
So what makes the bacteria different colors?
That's an excellent question. Different bacteria produce different pigments for different reasons. And as my PI likes to remind us, bacteria don't have eyes. So they're not looking at these chemicals the same way, like they're not seeing the red in the same way that I see the red. They're producing it for all sorts of reasons that include antibiotic resistance, UV, to fight against different stresses. There's a whole canopy of reasons why different bacteria make these different chemicals that we perceive as color. I think my favorite is that of Streptomyces species, because the pigments that they produce, pinks, blues, so many really cool colors, also function as their antibiotics. So that's why I'm really interested in continuing to work with Streptomyces species, including with collaborators in the Netherlands, because they know a lot about Streptomyces. And what's really cool about Streptomyces, I know I'm just sort of like getting really excited about that, is they have a chemical in them that's responsible for the smell that we smell after a rainfall. So that earthy dirt smell, that's like, oh, I love the smell after a rainfall, that's actually Streptomyces bacteria producing the chemical jimicin. It's pretty cool. Alright, are there any other questions? Any more designs that I could draw?
We have a design and a question. Can you show us those pigments?
Can I show you these better? Yeah. Here's the one that I was talking about, is still unidentified. I think because of how motile it is, you can see it's really, really slimy and gross. And some other properties they uncovered in the lab, that it's a pathogen. And this is Pseudomonas aeruginosa. This is a bacteria that my colleague Melissa Walker is working on. It's responsible for a lot of infections, a lot of hospital infections. So she's trying to figure out how bacteriophage, which is a special type of virus that infect bacteria, how they can calm these biofilm communities and reduce them so that people have less infections. There's also some application for Navy boats. She's doing a lot with this little dude. I also have some serratia, but it didn't grow up with wood. There's a design? I can definitely do that.
I am probably not going to say this right. She said, can you draw Dupui? D-U-P-U-Y.
I don't know what that is. But I certainly can look that up and draw it and give it to her. I would also like to say, okay, so I talked about how we do this as an activity in the class. In 2015, the American Society for Microbiology put out a call for more scientists and artists to create these types of living artworks. And my boy Alexander Fleming, back in the 40s, remember him? He was actually one of the first people to do this, so it's really cool how they're sort of carrying on the torch. But in our classes, we don't just use this as a really fun activity, and don't get me wrong, it's really fun, I really like doing it. But when this grows up, we're going to see really unique interactions between Pseudomonas and this bacteria that include antibiotics, different types of competitions between the bacteria. We could even see the red and the green combining colors, maybe looking like a brown. There's a lot of ways that this artwork can inform discovery, so we have our students look at the plates and create experiments based on those observations, giving them a little bit more autonomy in the classroom. The latest winner, if you want to look up the American Society for Microbiology's Agar Art Contest, the winner from last year created a really beautiful portrait of two feminine faces looking at each other, and one of them was really colorful, and it produced antibiotics that wiped out the color of the other feminine face. So I think that's a really perfect example of some of the same observations that our students are making in classes. And I think it's a really great tool also for the public to see antibiotic resistance and other types of microbial interactions in real life.
Okay, so Tandy Petrov's biology class is watching. Woohoo!
Cool. Hi, Tandy.
And she said what she wanted you to draw is her school. So it's the name of her school for her students.
D-U-P-U-I?
D-U-P-U-Y. That's what she wants you to draw.
Sure. Yeah. Let's get that down. Capital letters, lowercase letters?
Capital T, the rest lowercase.
All right. Let's do this. D.
She said any letter.
Any letters? If you can kind of see that. So it will grow up in about 48 hours. They take at least 2 days, these ones do. And I'll store them at room temperature. And we always invert our plates so that the condensation that's at the top of the plate won't affect the bacteria. Right. Any other requests or questions? Bacteria are really cool. I don't know, next time, what did my colleague Julian Jackson say when he was a graduate student here? Every time you breathe, you should thank a microbe because they do create a lot of the oxygen we breathe. Also, they're super cool because we breathe oxygen, but some bacteria, they can breathe metal. Like, they're really cool. Bacteria are very diverse, very dynamic creatures. We tend to think of them as just sort of circles or rods, but they have more complex shapes and more complex sizes. Bacteria are the coolest.
Well, thank you all for joining us today. Our next one's going to be September 25th. We've got 2 students from developmental psychology who will be explaining the dangers of texting and driving.
Cool, I should definitely watch that one. Well, thank you so much for tuning in.